Okay, perfect. Welcome, everyone. This is our first virtual biology community meetup. I'm very happy to introduce our speakers today. So the setup will be that we start with 3 presentations from Fabian, Ron, and Amaro, and after that we will have a nice discussion. In terms of questions, you can submit any questions anytime in the Q&A, and our team and also the presenters will try to tackle them in the meantime. And if you are voting on the questions, the upvoted ones will make it to the panel discussion. In terms of very quick kickoff, we felt that last year was really the first year of virtual cells. We have seen many great initiatives like the Virtual Cell Challenge, also new papers coming out from various teams. But we really hope that next year, or like this year, will be the year when we start deploying these technologies and pharma will and biotech will start benefiting from what we are building. So today we would like to share challenges, learnings, and basically an outlook for this year and the coming years for virtual biology. And we will start that by first Fabian sharing his thoughts. Also, please introduce yourself in a couple sentences before you kick off.
Hey guys, thanks for having me. Thanks for interest in this. I'll quickly share my slides and happy to introduce myself as well. Fabian Theis, doing computational biology and AI for single cell modeling. Very much looking forward to the discussion in a minute, particularly with my colleagues. Interesting to see what, for example, Noetic and Turbine and so on is up to. It's going to be nice. I'll briefly give an overview of a few things, what we've been doing in the past towards sort of how we build these virtual cells. I won't actually show much about the perturbation modeling, but I want to sort of show you a bit on sort of the embedding part, okay? And I kind of like this part on maybe sort of the one thing I want to stress here is this actionable models, which I think is something that you just don't want to only work with in a cell line, but maybe in some more complex setting. So what do we want to do? I mean, I think this whole concept of building cells or sort of using Gen AI to sort of see where we go, it's essentially that we want to sort of understand the cell landscape. So this was some older perspective that Nikolaus, many of us wrote, where you sort of maybe have cells going through transitions, at some point going bad things. What you actually want to do with them is you want to sort of learn this draw landscape here, which we see in parts in our UMAPs and high dimensional stuff, but we also want to act on it. So not just sort of get the position where we are, but also an idea where we would move if we did A, B, or C to the cell. Okay, so we want to get X, and we want to model this sort of observation, maybe just from RNA, from additional positions, but we also want to get this in a sort of time-resolved fashion. You know, we can reconstruct these trajectories because we have dynamics, we have pseudo-time, we have all kind of sort of much more complex things to get these lineages branching and so on. So we want to actually get to this sort of dynamic process. And in a sense, this could in parts already mean what you want to have a simulation model of a cell, which, you know, sort of what we essentially mean by a virtual cell to have. It allows you to predict how it would evolve over time. But in addition, of course, that's a key thing. You also want to add perturbation to that. So once, maybe in this case, just this example, you sort of see branching off at a disease trajectory, you want to go back. But there's many other ways how you want to sort of influence cells in terms of the perturbation behavior. It could be some sort of naturally occurring CRISPR, such a naturally occurring SNP, but it could be maybe also sort of exactly deliberately made CRISPR. You want to program cells, things like that. So this is what we want to do. And this is sort of what we started out quite some time ago. So early on, you know, we had in our first, I think we called it SC-Gen, something like 5 or 6 years ago, where we essentially had sort of cells under perturbation. This initial case, we called it drugs, right? And we sort of subtracted the ground state, added some new cell types, and we sort of generated these things. This was sort of one of our very early approaches for that, and maybe even now a quite popular baseline to sort of see how, with in this case, just sort of simple latent space arithmetics, you could already predict where things go. And even nowadays, there's a few papers saying that for maybe more complex things, additive perturbation is the way to go. How do we do all of this? And obviously, you don't need to tell this here, but you know, sort of with sort of deep representation learnings, let it be autoencode or more complex things. We just don't reconstruct data, but we also represent them. And you know, these representations with, for example, VAEs and so on, are really powerful and effective. And you know, this is sort of introduced the normal autoencoders from my lab, VAEs with NIRS, and now sort of being really one of the very popular early ones that we sort of since then have extended in various versions. All right. The cool thing, what you can do with them is also sort of add additional phenotypes. For example, we have many of these. You can use them for integration, for learning atlases, and so on. And this is what's been really a powerful thing for us to describe, not the way where we move, but as a first approximation to sort of cell state space learning where we are. So this is this whole atlas learning. And this is just one example of these atlases. This was work by Leander in collaboration with Cizong from Barbara Teutlin's lab, where we learned the neural organoid atlas, where we had like all these different data sets with sort of developmental behavior. So this, this sort of this X of T was quite well defined in many things. And we just integrated within a sort of a big map that allowed you now to map all of these organoids to a joint thing, and you could sort of see where they would go, where sort of they differ from maybe the different protocols and observations that you had. But, and I think this is kind of cool, then you can actually map this to the primary case, because now that you have sort of a sort of somewhat usable object, you can actually see where it would differ in this case, we had a developmental, like a fetal brain atlas in the human from Stan's lab. And then you can see, hey, some of these, I think, blue ones are where sort of the organoids map very well, but the red ones are just sort of not hit by any organoid protocol. So they're depleted. And now, of course, you can ask, hey, if my organoids make all of that, like, how can I make this stuff? And for example, this stuff I can't make because these are immune cells, typically hard to put into organoids, right? But then there's a whole bunch of other cells and these sort of your low presence scores things, these ones here, and they sort of are typically underrepresented cells in hindbrain, midbrain, and so on. And now, and that's sort of a virtual cell challenge, if you want. How can I now change my experimental space to make these things? This is sort of what you want to use models for. And this is not what you can do with these ones. These are not causal, right? So I think where we are is we can sort of build these reference atlases, but we cannot, we just have data, but we know we don't have these models. These are sort of these examples, these pictures that you might have seen from CCI. Hey, let's build these virtual cells. And by the way, from our this discussion earlier, Amaro, this is not last year's virtual cell year. This has been in discussion for quite a bit longer, right? I mean, we had CCI things and many others for a long time. But the idea that this gets now, I think, is really getting in the broad public now. Actually, it's an old thing. Like acting on cell landscapes, this is not our idea or even idea 5 years ago. This is old stuff. Systems biology has been trying this for more than 20 years. If ever, whenever we sort of the genome has been sequenced, people have been thinking, hey, now we have all these bits and pieces. We have metabolic dynamics. We know how stochastic variation we see across cells. Can we build these whole cell models? Yeah, and then they had these type of dynamics and were not very predictive. So, we need to do more. So even though people, hey, do you see this? 2012 spoke about virtual cell biology, an older term. But I think, and I'm simplifying, of course, I would say none of that was actionable. This was really not like how we behave in a generative new situation. But people have been thinking about it, asking questions, and you, Demis, for example, talking about virtual cell as a bottom-up thing built sort of out of proteins, AlphaFold style. Or, Priscilla Chan and Mark Zuckerberg in this sort of CCI foundation really putting a lot of effort there, and many of us thinking about this. And here's one sketch that I'm sure many of you have seen. This was written by Charlotte with many of us, particularly as Steve and Emma, sort of pushing for this as sort of this big concept where we go beyond just this sort of integrating and sort of melding and making references, but sort of also sort of seeing in this virtual latent space how perturbation effects shift stuff. This was this plus minus thing that I showed you before. Different perturbations, maybe even sort of fine representations. All right. So that's the idea. I think at the core of it lies, of course, that nowadays we know how to build these foundational representations of cells. We suck up a lot of data, maybe across different data modalities. This is from a perspective that we recently written. And it could be autoencoders, but of course nowadays the way to go typically would be transformers, even though I still argue the encoding is not fully solved and we're still sort of playing around with what type of masking and so on works. We had a recent paper on different ways how we can, for example, do masking. And I think it's so that there's I think a lot of things to fine tune. But in any case, once you have such a representation, the key thing is you just train this once, you ask a whole bunch of questions downstream. And I would say, I don't know, Mara, what you would say to this, but this I would call already a virtual cell because you can curate it with different things, potentially fine tune for that. A seminal paper that sort of kicked all of that idea off, it was a Gene Former paper some time ago, and since then many modifications towards that from us. Also some things, but there has been some sobering up. This concept sounds powerful, but for perturbation models, the perturbation prediction task, the plus minus thing that I showed you before, maybe didn't do even so well. This was just a recent paper from Wolfgang's lab, and where they showed that additive things sort of don't. Where was it? I think here prediction error. So just additive baseline is hard to beat. We've since then found some examples where we see really non-additive effects, and you need to go into basically more complex stuff than cell lines. But still, classical stuff didn't work so well. And we had even some older papers where even just these deep embeddings might not be necessary for simple tasks such as cell annotation. Cell annotation is such a popular task still to evaluate models. Nah, just logistic regression, fine for most of these. In any case, we asked some more complex tasks. The other day, we had a spatial omics foundation model where we actually had spatial context. You want to impute from a certain cell what its neighborhood looks like, and then, you get to sort of maybe more challenging areas. But let me maybe show you one thing where I think we can really change something on the encoding. And this is, I think, maybe a bit timely because, the field is sort of asking itself, what do we need to do? Yeah, with many of these competitions, just not exactly going there. So could we do maybe something different? And I think some issue of maybe those current approaches, how we embed them, is that this mass modeling just looks on the reconstruction, and maybe we don't even care always about reconstruction. In the end, of course, we want to see what those new generated behavior is in actual gene or some other space, but maybe for sort of effect, it's good first to look just in latent space. And you're sort of going beyond generative architectures where a lot of effort is being put on reconstruction. For example, Jan recently, and he has been sort of going for this for a long time, talking, hey, we actually need maybe better representations, not just reconstructions. So he's been challenging the whole masked vision transformer type of approach, and there could be maybe more to it. So in his case, he's been talking a lot about JEPA. There's some first single cell versions of that, where essentially sort of instead of reconstructing, what you would do, so after decoding, you just go in the latent space and you measure the distance of two sort of related embeddings. We've been trying something a bit simpler, just based on a simpler version, essentially contrastive learning, but this actually gave us quite some nice things. This is work by Mushtaba. We call it, I think, SE concept. The idea it would be that we take two different views of cells and then compare them whether they're same or not, and thereby sort of learn in good embedding. I just show you a few results of that. This is going to be short talks, but I just want to have something sort of a bit more content rich before I then go to outlook and representation. So the idea of the method of SE contrast that we have sort of this pre-training that really tries to produce consistent cell embeddings, not reconstructions. Okay, so this is sort of the challenge that we do. In order to make it vary, if we just have RNA, we just sort of split it up into different views. If you have RNA and other modality, you can sort of use that, right? So here, for example, you would just sort of discretely sample for two different panels, and you would ask, hey, are these now coming from the same cell or not? And sometimes you can tell, sometimes not, right? But this is sort of what you can input. Now you need to encode them, and for this to be thrown into a transformer, you need to sort of do some tokenization. There's various ways how to do it. We take the typical common one now with respect to gene ranking by expression and sort of adding also position encoding. There's other ways how to do it, and we've been benchmarking them. For us at the moment, this was working best. Then we would take the typical transformer type of encoding. You can play around with the scales here, but what you would get is sort of you get two different cell embeddings of those two different panels. And I don't think I need to show what the transformer would do. So now you have the embeddings, and with those embeddings, what you would do now, you would sort of take a similarity matrix of the two, and of course, these things we know that they would work out, right? But the question is now, hey, how about the comparison of panel 1 to panel 2? Like, how they're similar or not? And for that now, you put the typical info and C loss from contrastive learning. So sort of make close ones move closer, make different ones spread out more. And this is what you would actually do here. And it seems to be doing something. So let me show you first how the training works. So we take sufficiently large model, and there's again a whole bunch of choices. We really benchmark quite a few of these things, but overall we need then maybe just something like on a bunch of GPUs, just for the final thing, just train a few days. We can upscale this and add more data. Is currently what's happening. But I think what's interesting is that, and this was an issue with early on foundation models. We never saw this scaling behavior. A key concept in foundational LLMs and so on, right? Where you showed, hey, the more data, the better it gets. In this case, sort of the more parameter, the higher the accuracy. So we need apparently this model complexity. We also showed that the more genes you add, the matching accuracy goes up. So it seems that, this model is able to suck up the complexity of the data and actually do something interesting with it. And now I have a bunch of examples of what it could do. For example, here, we can now use this for this very simple task of cell representation. Maybe not as impressive, but definitely outperforming simpler models, ours included. And in particular, if you fine tune, it gets on par with sort of specialized methods. In this case, just an simple logistic regression, which typically outperform those classical type of general models much before, and now sort of really going on par. And you actually also see that they nicely integrate much more so than others. And interesting now, we can sort of query a brain reference, a brain samples with we have a sort of a healthy reference, and we query MS patients, and we see that the fine-tuned version, even the normal ones are already doing well, but the fine-tuned version even outperforming sort of more classical approaches. And this works also for other data sets. And here you interestingly see that sort of by keeping on training and adding more data, also you sort of really get beyond sort of the thresholds of simpler methods. So that was really nice. And because this model encodes the genes, we can actually, without reconstruction, even curate what sort of the key gene factors are that sort of push through this, and they sort of match up with questions what you would ask for DEGs. All right, so annotation, maybe not the most exciting thing, but now you can actually do cross-technology label transfer, right? Because this thing sort of wants to learn a robust embedding. So let's take whole transcriptome, a subsetted xenium panel, and then we can see how well they co-embed. And then they co-embed much nicer than, the simpler previous foundation models, unfortunately also ours included. So this seems to be doing a good job. And, we can also see how much the drop in terms of performance is when you go from the full panel to this targeted panel, and it's much higher from existing ones, including also linear methods such as PCA, which is sort of typically harder to outperform versus others. We can look what's included, and I think I'm going to skip this, but I think what's interesting is we can do some cross-technology label transfer. So let's really try this. Let's take a single-cell RNA-seq data set and try to annotate with these ones a spatial reference. And people who have tried this will notice that, there seems to be some dynamical shift. So just subsetting this to the gene panel doesn't suffice. There's sort of more happening here because, typically the sequencing-based technology is different than the fish-based one that's been using for readouts here in typical cell-based assays. So what you would do now, you take an ovarian cancer reference and you annotate now a xenium a tissue panel, one that actually has sort of, in this case, a lot of observations. And you see that with the normal one, we actually outperform quite a bit many of the existing sort of simpler foundation models, but with the fine-tuned one really goes beyond and goes into regions where this is really becoming very useful. And we can use it now for imputation, right? So let's take a reference and impute on the spatial one a single-cell RNA-seq. Let's just first do it on a holdout so that we can measure it. And we see that, in terms of accuracy, this is doing a good job, better than others. And it sort of works well for Murphy's sample. And we can essentially now, with this, of course, use it also for panel optimization. As a last thing, there's sort of other things that you can do with such a potentially flexible model. For example, let's say you have fixed already your single-cell transcriptome atlas. You may have gone for community annotation. This is typically what we do within the human cell atlas. So what would you now do with such an annotation where you can sort of retrain, and you sort of don't want to sort of retrain a foundation model on that? Or you can now do a projection, a very common task that we do. So you can now use this one to map basically spatial atlases to existing atlases, right? So can you sort of embed this for your lung cell atlas, and you can show that, sort of you nicely are able to map, in this case, for example, epithelium cells, and you can show that this label transfer is working robustly. You can even quantify this, that this sort of outperforms to quite some extent our previous sort of methods where we just keep the structure of the variation autoencoder type of fixed. This is what we call SCR just some time ago. Last thing, you can also use this for integration, the powerful foundation model. So this is sort of all virtual cell tasks in terms of sort of what we listed before. I won't show you perturbation, this is going to say in a minute. But for example, with SC Concept Plus, we can actually sort of really map liver samples into breast. I think we mapped ovarian cancer into, sorry, I need to look up what we did here. But in any case, we saw some mixing here, and we can quantify this sort of generation of sample mapping, and you can see sort of that this integration seems to be doing a better job than previous foundation model and zero shot, but also domain adaptation, quite strongly outperforming these. Okay, with this, let me just sort of take on orthogonal route. Sort of a parallel thing that people have been thinking about instead of just going very specific model as we did here is, hey, can we maybe in parallel just use language? Won't that maybe not be enough? And that was this really, initially, I thought, man, is this really working out? Paper from David called Cell to Sentence, and then from Christoph Borch's lab, the Cell Whisperer, where essentially they used language models, sort of encoded the gene expression as text and then co-embedded. And it seemed to be doing something, right? There's a whole bunch of very interesting tasks that they seem to be able to do with that. So like what's contained in the structure of a language model that tells you already about gene relationships? Apparently something. So I wanted to study this. So in our lab, we in collaboration with colleagues from Shanghai, we came up with what we call Cell Hermes now, where we essentially fine-tune a natural language processing LLM with essentially sort of adequate question answering pairs. And we can do some things that seem to be impressive. So very interesting to see which sort of routes full, so just database pre-training or even using language is helping, or maybe sort of it's going to be sort of composed just with orchestrating agents. Interesting where things go, but I think just some questions we can ask you can say, if I have a cell that has sort of these genes expressed, is this cell sort of reacting to something, and explain the reasoning, and then you can sort of get some explanations from this model. I think it's kind of powerful with respect to explainable AI. In any case, so this just for foundational representation, I think this contrastive pre-training can be a very effective way to go for this. In the last 3 minutes, let me just sort of go towards my outlook. A big task, of course, is not just sort of to get the X of t and where it would develop, but sort of how it behaves under perturbations. If you want to be sort of very strict about, and this was from a perspective that we did with Joshua some time ago, where we essentially ask, we have a whole bunch of perturbations. What would happen if we model this as an intervention? Could we find these causal mechanisms, right? And this is what you would do. You would sort of just compare then the measured outcome from the predicted and various assays, and then you can sort of build up models for that. And there's something, we have one method that's based on our Moscow paper, we call it cell flow, together with Greg Kemp's lab, where we have a flow matching type of approach that really enables us to sort of see where in a generative fashion cell perturbations could go. And this seems to be, for us, a very powerful way. There's a whole bunch of other flow matching papers coming up since, and I think there's quite some popularity of these ideas. I won't show you those results. I've been talking about this quite a few times before, but just want to show you what you could potentially do. Let's say we have now our multimodal foundation model. It could be a C-contrast one, could be others, could either many modalities, just single sign as seq or spatial omics. But once you do this, I think this idea of not executing these perturbations, this is something that we want to do with virtual cells, right? We want to do experimental planning in silico before we go into the dish, right? So we would sort of generate some hypotheses. This is what we do just completely in silico, but then we would sort of propose right experiments, and we sort of update the model iteratively. Classic idea, as we sort of outlined here, perspective with Bohm. And last but not least, what I think this is cool is this part here. This is not just perturbation prediction. If you really think about it, you don't just want to sort of completely explore perturbation space. You actually want to do some search in there. And search, if it's just yes, no type of perturbation, maybe you can just brute force it. But in practice, this is a hard problem. This is sort of what robotics people need to do when they explore the robot going through a maze, right? Like you want to eventually go somewhere. Like how do you do that? We've been coming up with some approaches to sort of solving these inverse models. We call it inverse cell flow. This is ongoing, so this is completely unpublished, but I'm super excited about these ideas and also results that we're having. Then let's say you have these three observed, you want to sort of go to this new population. What do you need to do? Well, this is what you need to do in permutation, in sort of, this would be cell space, this would be sort of perturbation space. So questions how to explore perturbation space, I think we're going to see a lot of these in the future. So with this, let me wrap. I think cellular foundation models, there's a whole bunch of challenges. We need to look for computation. We need to look for evaluations, a really important thing, ethical considerations, and also sort of this forward-looking perspective. Just want to say one thing about evaluations. There's been so many models, so many ideas coming out in the field that you may need to somehow agree on some evaluations. We've been coming up with this open problems thing in a big consortium and group of colleagues that just came out some time ago. It's this website where you can come up with problems and then you can sort of see how different methods perform. I think it could be a very nice independent way. There's, of course, also a whole bunch of challenges on Kaggle and so on. One from the ARC Institute, which maybe a bit over-promisingly was called virtual cell, de facto was a perturbation challenge, right? But I think also there are some problems you see with metrics. Here, basically, we allow all kind of metrics, but there was some sort of quite simple, I think, early on obvious sort of metric hacking where metric happening where essentially the top many results weren't entirely exciting. And then I think result 201 or something like that, which de facto was actually flow matching, right? So very excited to see where things go. I think very important to have such data sets, but we need to compare. Yeah, I look forward to the discussion now. This was my lab. I think I acknowledged most of the people and I look forward to discussion later. Thank you.
Thank you, Fabian, for the excellent overview. You really gave us an overview from like also showing SCGPT and all the beginnings to where you are today, and it's super exciting. I have many questions to ask over the panel. I actually want to encourage the attendees as well that in the Q&A you can ask questions. I haven't seen one, so please go ahead and ask questions. There is one. Okay, good example. Follow that example, and please, Ron, come on board and show your presentation next.
Great. Are you seeing my slides, everyone?
Yes.
Great. Great to meet you all. Thank you all for being here. A lot of folks attended. This is awesome. So I'll be, relatively brief here. I just want to give a little bit of an overview of really the company and then how we're using, some of the models we're developing. Some of the things I'll preview, we're about to release some longer write-ups, so you can sort of stay tuned for some of this. And I don't want to exactly steal our own thunder, but we'll preview some of the cool things coming out. So really, Noetic is focused on building foundation models of human biology. We have done some work around the virtual cell model space, and I'll share some of that. But really, our thesis is how do we build the right data sets and then train the right types of models to solve a lot of the therapeutically relevant biology problems, especially in a regime where we are getting better and better at, the drug design problem. So that's really our focus is, a diversity of different applications there. The main thesis for the company has been that, the problem we face in making new medicines is clinical success. So, everyone hears the statistic that 90% of drugs fail in the clinic. That's absolutely true. And we think a lot of that failure is not because we haven't necessarily designed the molecule right or the molecule is not safe for humans, especially at this point over the years as we've sort of accumulated a very large stack of de-risking getting into the clinic. We think much of this problem is a lack of understanding between the connectivity of the preclinical work, so the work we're doing in animal models, cell cultures, systems, et cetera, and then the work we're going to do in the clinic. So which patients are going to respond to a drug given our results early on in the process. And so what we said is if we can begin to train models on exactly the types of data that are going to be relevant for the clinical problem, so that is patient tissues, then perhaps we can begin to solve this translational bottleneck and increase the probability of success downstream as we get to the clinic. So really that's our guiding principle, and a lot of what we do is how do we understand the connection between a particular drug and the right patient population. Now, to do this, we needed to start with the data. And so, we didn't have any biases. So, I came from, Recursion. I was there for 6 years from, very early days. And personally, I'm not someone that's been in the single cell RNA-seq space. So really, we approached this problem from first principles, and we said, okay, if we want to build this data set that is going to be human relevant, what would it be? And spend some time sort of thinking about that. And ultimately, we decided, okay, we're going to have to build a lab. We're going to have to source human tissues. So in our lab in South San Francisco, essentially, we are bringing in commercial tissues, commercially available tissues. So these are human tumor specimens from a diversity of tumors across all of oncology. And then we process them through essentially a workflow that we aim to generate what we think of as a fit-for-purpose data set for machine learning. So what that means is, a lot of the same principles that we applied for the in vitro experiments, phenomap at Recursion, begin to apply to this type of data set. But in this case, we're not looking at in vitro experiments. These are actually human tissues. So we try to control for things like batch effects and other types of artifacts that are going to be a problem downstream on the machine learning side. Ultimately, at the time we started the company, we felt this is the right moment to begin to use a type of data modality that had just come online, and that's spatial. And so a core to everything that we're doing is essentially 4 different types of data. So we're generating for every tissue, standard pathology H&E. A multiplex protein. So the protein lets us look at the different cell types within a tumor. So the tumor, the immune cells, TME. And so we go from sort of the tissue level to the cellular level, and then spatial transcriptomics. And today we're generating whole transcriptome, so 19,000 genes. And you can see here, we're not taking just a small window of spatial transcriptomics. We put all our samples on tissue arrays, and we're essentially covering every square inch of every sample with FOVs to capture all of that data. What's very unique about our data set, in comparison to a lot of data sets out there, is everything's paired. And so, for example, the H&E and the protein are on one slide, and then 4 microns away on the serial section, we're collecting spatial transcriptomics. And so the model is able to learn from essentially the same cells. So the model sees H&E and protein, and then in many cases for the same cells, it can see the transcriptome. And so it turns out that that is a really powerful benefit in beginning to train these models, because you can start to move between these modalities. And it's actually quite challenging generally to train multimodal models when a bunch of your data is in modality 1, and then you don't have paired data in modality 2. You're sort of bringing in a disparate data set. And then the 4th modality here is we have we do genotyping of all our samples, and that's standard oncology genotyping panel. And so essentially here, that's our workflow. So we train a number of different models. These are all transformers based and have different applications, and I'll show you some of the downstream. But really, what we tried to do is use different Fabian really described all this quite well. So, different types of masking policies and different types of approaches to think about how we can get these models to learn from all the different data modalities. And certainly, tokenization and the nuances of masking policy are important here. And some of the key benefits of the key drivers of different model performance. And so we haven't really talked a lot about the specifics of any of this, so I'll brush over it a little bit and just get to applications. But we are, suffice to say, we are seeing really exciting progress on a lot of these different approaches. So the other key thing to think about in this space is, these data are huge, and you can begin to scale, build quite large models that are quite expensive. So I'll show you a little bit of work in the last couple slides on scaling, but we are currently working on very much working on figuring out how we can begin to scale these models, and we are currently seeing performance increases from scaling these models. So the largest model we've trained at this point is about 4 billion parameters on about 320 H100s over the course of some weeks. That's quite expensive. We can't do that very often. We are not exactly funded like a lot of the frontier AI labs. But we think this is really just kind of the beginning of what these models can do. So now I'll just go through a couple different applications here. Some of these are discussed on our technical reports and in our blogs, and then some of them will be coming up soon. So maybe the first one, most relevant here, is the virtual cell model. So why do we think of this as a virtual cell model? So really, we're not trying to model like a cell as a bag of chemicals as such and understand all the biochemical reactions. What we think of as a virtual cell model is this model that's actually simulating is virtualizing a cell, and then that's the use of the model to do biology. And so what I'm showing you here in this movie is a piece of a patient tumor sample here on the left, and what you're looking at is in magenta you see the tumor, and in yellow you see the T cells. And so what this model is going to do is simulate the placement of a T cell, a virtual T cell, at any position that we specify in this patient tumor. And based on the positioning of that tumor and the spatial context and the surrounding biology, the model can infer differences in underlying biology. And so you can see that here in this middle panel. And so I'll just rerun this movie. And so what we're going to do here is we can actually prompt this model. So here we're prompting it with a set of genes, CD3 and CD8, that's specifying that the cell that we want to simulate, the virtual cell, is a CD8 T cell. But you can imagine prompting this model with any genes of interest to get to other types of cells or subtypes of cells. And so what this model is doing is now inferring the gene expression for this virtual T cell across this tumor. And one of the things you really notice, one thing in particular, if we look at IL-7 receptor, which is a naive T cell gene, there's a strong differential expression or inferred expression, as it were, for this gene based on whether the T cell, the virtual T cell, is in sort of this nest of other T cells here at the top left, or whether our virtual T cell is in the middle of the tumor. And so what this tells you is, spatial context, very much matters to understand the biology of T cells. And you can do this simulation across, other different cell types. And we can also do sort of more complex simulations. So here, what we're doing is we're simulating, suppression of a drug target and asking what happens to, other downstream pathways. And so what we're doing is essentially prompting the model and saying, okay, give us sort of the end result of the transcriptomics across different conditions, and those conditions being the gradual suppression of VEGF transcript. And so essentially we're virtually knocking out VEGF, or knocking down, as it were, VEGF signaling in the tumor. And then we're asking the model to infer an output for each of those. And the model will make that inference across the whole transcriptome. We can even do it across the other modalities. So you can do this within the protein layers, but largely we tend to do this with the transcriptomics. And you can begin to ask questions, what would happen, in a particular tumor if I were to change the expression of this drug target? So these are all just kind of nuts and bolts examples, like the engine, running some of these simulations in a single tumor. And that's interesting, but really what you want to do is be able to scale these simulations across hundreds of patients. And so this is these are some results that we shared in a poster at SITC, I think a couple of years ago. So this is also online on our website. And so what you can do is run these virtual cell experiments across hundreds of patients in the cohort. And so, for example, we have over 1,000 non-small cell lung cancer patients. Each of those patients is represented by dozens and dozens of individual tumor cores. And so what we can do is say, okay, well, let's place these virtual T cells across two different cohorts of patients that we know are interesting. In this case, we're looking at a set of patients that respond to immune checkpoint inhibitors really well, which is these KRAS patients, and a set of patients that don't respond to KRAS to checkpoint inhibitors, which are these patients that carry these STK11 mutations. And so essentially what we're asking is, okay, if we put virtual T cells across all of these patients, what is the differences in T cell gene expression across those different cohorts? And then we find some genes of interest that we can then use as queries to knock down. And in this case, what we identified were several granzymes were differentially virtually expressed between these two cohorts, and then we can virtually knock down granzymes. Or we can virtually knock down genes across the genome and look at which ones will, hopefully increase the granzymes in the T cells in the non-responsive groups. And so that essentially becomes a virtual drug screen where you've identified some biology, and then now you can screen against that biology. And it's completely in silico, and it's all derived from these human tumor samples. And so the hard thing becomes like, how many of these experiments do I want to run, and how can I set these up and run the inference, basically. And this is really why, in the past I've said, okay, a lot of what we think about is moving, the biology from like essentially the wet lab to the GPU cluster, because now you can, you can run all, the entirety of this experiment in a simulated mode. And then, obviously downstream, you want to understand whether your targets of interest are relevant or not. And so you can do some downstream biology. So, those are virtual experiments. That's in virtual screens. You can use the same model. Again, this is OctoVC here. You could use the same model to describe patients at a much larger level. And so this embedding space essentially represents an approach where we run a series of virtual cell experiments across multiple different cell types against every patient, and then we can concatenate that data into basically a profile of every patient. And so you could think of this um, this representation space as every patient, essentially profiled by many of these virtual cell prompts to get to these different representations. And so this is, again, the models are completely trained self-supervised. We don't have any labels here on treatment response. So the model is essentially like learning patient biology from these virtual cell experiments. And then we can go back and ask, okay, well, we have these groups. This is a simple latent cluster here. We have these groups, are these groups interesting? Is there interesting biology here that the model's finding? Does it make sense? And it turns out, for example, what I'm showing you here is this group in the top of this plot in gray. These patients tend to, if we look at our actual response data for, other patients that are not in the training, we find that the patients that respond to immune checkpoint inhibitors cluster very nicely with this gray group. And so the model has learned, a set of patient biology that corresponds quite well to immune checkpoint inhibitor response. And because we use the virtual cell experiment to characterize as sort of the underlying feature set for this, we can ask, well, what are the features that underlie these patients in gray? And it turns out that the important features are the things we know about for these patients. So these tumors are T cell infiltrated, they have high interferon gamma, high antigen presentation, et cetera. And we are not super interested in these patients. Obviously, these patients are well accommodated in terms of therapeutics, but we are especially interested in some of these other groups. So for example, as you start to move away from this gray population, many of these other groups have not really been described, and there are underlying biological features that can lead to new therapeutics that we just haven't discovered yet. And so that becomes, the challenge here is, okay, if we take this group in the red, what are the features that underlie this biology, and how do we identify therapeutics there? And then you can also use this type of approach to essentially begin to understand whether a particular drug may or may not work in the clinic. This is sort of a retrospective example for a program run by Merck and Daiichi, which was a HER3 ADC. And so what we wanted to know was, if we were to, if we had a HER3 ADC and we were to run it across our lung cancer patients in sort of the same clinical format, what would be our predicted, outcome, and would it be similar to what they saw? And so what we can do with this model is we can ask for, where the virtual HER3 expression would be across all of these patients. And get sort of a density of virtual HER3 expression, which is shown here in the middle. And it turns out that this particular program was run in an EGFR selected population. And so then we can ask about the intersection of EGFR with our HER3 virtual expression. It turns out that, if we sort of simulate their trial, then we tend to get a very similar result in terms of our simulated HER3. In the EGFR population. But really what we'd like to do is begin to increase the overall response rate of these types of trials. And so what we can do is use these types of models to say, well, if I need to enroll a patient population where I'm going to get an ORR above, let's say, 40%, what population would I choose? Would I use EGFR as an inclusion criteria? Probably not. So that's that. All of that is the virtual cell model biology. Increasingly, and we're going to release some more work here in a few weeks. So one of the next approaches we're taking is a model that is slightly different. Doesn't operate on the basis of the cell, but really is working at the level of the data in a more raw format. But really leverages the fact that we have highly aligned H&E and spatial transcriptomics. And so what this model is able to do is takes in as input both of these modalities, and what this model is able to do is infer the spatial transcriptomics from the H&E alone. And so we can do this across the whole transcriptome and roughly 19,000 genes. Many of these genes in the whole transcriptome refer to genes in many tissues that are not represented in our data, for example. neuro, et cetera, all of our data presently, apart from some normal tissues, some fibrotic lung, are tumor samples. But what we can see is that using this model, we get a very good prediction accuracy across a large proportion of genes. We think this will continue to increase as we get more diversity in the data set. And then generally, as one would expect, we see that the prediction accuracy varies with the representation of that gene in the actual data. And so really, so then you can begin to use this model to work from H&E alone and infer spatial transcriptomics. And so what we do with this model is we're really excited to use this model, directly on clinical samples where we can bring in biopsies from a clinical trial. And in this case, these are biopsies where we're not training the model on them. We're essentially running inference. So we get in a biopsy, or in many cases, we just get in an image, an H&E image, and then we run inference to get from that image to spatial transcriptomics. And then we can use this model to characterize the patient population. And ideally, our downstream goal is to train a simulator that would tell us whether these patients would respond to that particular therapeutic or not. And so we've done this across, probably about half a dozen programs right now. And in many cases, we can see, quite good results with even a small number of patients. And so here's an example of where we're, testing. Essentially, we can train a classifier on the model embeddings, and we're basically doing we're doing validation to test how well this classifier works on a patient-by-patient basis by doing data splits and asking how well the model can predict responders. And this is not a huge amount of data that we have for some of these trials. These are relatively smallish trials, but trials where we have patients that are responders and non-responders. And for this particular one, we get an area under the ROC curve of about 0.8. And so really our goal here, from a therapeutics perspective, is to look at programs where there was, an actual clinical outcome that was insufficient. And so that's what's shown here on the left. So maybe the clinical trial had some patients that very clearly benefited from this therapeutic, but it was roughly 20% of the trial enrollment population, but very clear signal, and ask, is there a patient population where if we re-enroll that trial, we can get to a larger response. And in this plot on here, we're talking about 80%, but certainly you don't need to get to 80% clinical trial setting. We can identify populations where maybe you're moving the response rate above 50%. And our hope is that will allow us to better enroll essentially the right drug in each, or the right patients in each trial for a therapeutic. This is just a preview of what's coming, so I'm not going to say much about this, but you can run this H&E to cosmics inference model on other tissues that are not human tissues, and it's quite fascinating to think about the outcomes of this. And so we will release more on this system, in some weeks, but we're calling this multi-species alignment and reasoning on spatial data, and really trying to think through the implications of humanizing model systems in silico. Lastly, just a couple slides, and I think we'll push this work out in either later this week or next week. As I mentioned, one of the things we're very interested in is model scaling, and understanding how we can get to better performance of these models. And what we've begun to appreciate is that the thing that's really important for these types of models is context. And so context, in the context of what we're doing, refers to how large a patch of the data the models are seeing. And so you could think of training on a relatively small patch where maybe the model's only seeing a handful of cells in a spatial setting, or increasing that to the point where the model is able to see the entirety of the tumor. Now, the challenge in bringing in these larger contexts is, these are very large data, and obviously this is a on GPU memory problem. And so what we've been doing is asking, how does context impact improvement in model performance? And so far, what we've seen, and these are early days here, but what we've begun to see is that if you as you increase the context window for a particular model, larger models learn better. And so the context matters more for larger parameter models. So for example, here we're saying that the 4 billion parameter model benefits much more from a larger context than let's say the 20 million parameter model. And so this is we're just starting to get to sort of the steep end of this curve here. And so really what we're trying to do is begin to push methods out to further increase the context length to the point where the model can see the whole tumor patch. And what this means practically is shown sort of here in this animation. And so if you look at here's an H&E, and what we're showing here is different regions that the model, semantic regions that the model is recognizing. And it turns out that in this particular tumor patch, there's an immune structure up here in the upper left corner, and then the tumor regions in the bottom right corner. And so you can see as you increase the context length, the model is able to better recognize biologically relevant information from the H&E. So on the left, you're really not getting a very strong representation of this tertiary lymphoid structure. Now, as you get to longer context, the model is nicely differentiating tertiary lymphoid structure as well as tumors in other regions. And so what we want is for these models to learn all these relevant biological contexts. I think this is my last slide. Yeah, we're excited to announce we just announced a partnership with GSK to work on OctoVC. I'm happy to answer any questions about that, but that is my last slide. I'll end here and pass it on to the next presenter.
Thank you, Ron. Congrats on the deal. There will be some questions on that later because there are some people interested in business model and economics of virtual cells, so I will ask you. Also, there are questions to you in the Q&A, so if you can answer them in the meantime, that would be wonderful. And I need to have this joke that you may reach Mars earlier than Elon with your technology. That's exciting. Okay, Amaro, you come next. Thank you, Ron.
Okay, can everyone see my screen and hear me?
Yes, the screen is small, but...
Oh, sorry, hold on one sec. All right, hold on one sec. Is that better?
Yes, perfect. Thanks.
Okay, cool. Hey everyone, nice to be here. I'm Amaro. I'm the CTO and co-founder at a new company called Atlas Medicine. That's a multimodal precision medicine company. Today I'm going to be talking. My talk's a bit different. I guess I'm taking like it's more opinionated about like what's going wrong about like the field maybe. But this is work that I prepared with a colleague, Johnny Lee, who is him and I developed some of the model and the blog posts and things that we have been sharing in the past. So I'll just be giving an overview of that. So just starting off, just wanted to like position the way that I saw things unfold over the last couple of years. I would say that the impact of single cell foundation models has been relatively underwhelming for these kinds of models hitting a new area of biology. And I think there are several key challenges here. One of them is data, another one's evaluations, and the last is inductive biases in models. And I promise that Fabian and myself did not coordinate our slides, but we have, I think, similar takeaways. And if you look at like some of the papers that came out, you can see that there are several articles where they compare single cell virtual cell models or these foundation models to linear baselines, and the linear baselines tend to outperform. So I'll be talking about a little bit why I think that is. So in terms of data, I think one of the things that we've seen is that data has scaled a lot. We're seeing single cell foundation models with greater than 100 million cells. But in terms of the quality and density of that data, we tend to see sort of the same cell types in publicly available atlases represented many times. And there's relatively low amounts of data in tasks or local areas of this kind of like data landscape that matter for downstream tasks. So things like perturbation modeling, things like patient primary cells, you just don't have a lot of data in those areas, and that data is perhaps most useful for downstream tasks. So I would say that one reason that models may have been underperforming our expectations or under delivering in terms of impact is that we need to increase coverage in sort of the local manifolds of data that really matter. And Noetic and Ron's data really, I think, exemplifies what doing this well might look like. Um, One thing is I put this slide together before Illumina announced their 1 billion cell data set. I haven't taken a look at that. I think that came out yesterday. It should be really cool. Love to play with that, but it'll be interesting to see how that changes things. Another area, and this is something that I think practitioners in the field at this point are familiar with, is that I think the evals are just a poor proxy for performance in tasks that matter, like for example, for drug discovery or target discovery. So atlas scale tasks, these are the typical tasks that people measure the performance of their single cell foundation models and train for are relatively easy to learn, even for simple models, and involve this kind of like coarse variation. So like cell type annotation is a good example of this, where you have an atlas scale, you have all these different cell types, you want to just like label the individual clusters. PCA works really well. But for the tasks that really matter, there's small local shifts that are really important to capture. So like perturbation prediction or patient stratification involve really understanding some area of this overall atlas where the subtle signals are perhaps competing with the noise floor that comes from single cell sequencing as a technology. And then, I think this is a problem, and Fabian mentioned this as well, but, there's a real lack of consensus and single sort of spot for benchmarks and metrics. So, like in protein engineering, there's like the CASP challenge. And that's like that kind of like clear benchmark that everyone agrees upon and the academic community supports is really important, I think, for these benchmarks to really function. So, I would highlight that metrics for single cell data are really complicated. You have to think about like how many cells do you sample? How do you deal with dropout? And how do you deal with perturbations that maybe have no effect? Like how do you compute your metrics in that setting? And then I think a proliferation of benchmark tools without academic support and a real community behind them to evaluate them is a problem. So metrics, as we saw with the virtual cell competition, sorry, can contain sort of obvious flaws, and the field can't really catch them fast enough in order to get these things before they kind of propagate. And I think, and the DinoCell paper and companies in general are guilty of this, but preprints also contribute to this, right? Like if you don't go through review, maybe there's like metrics that aren't really useful. And this XKCD comic is always useful for this kind of idea. Okay,
Strongly
so
agree. Love that, man. I always use it a
uh,
lot. Sorry for jumping in. I just really love it.
oh yeah, no problem. I mean, yeah, we didn't coordinate, but I feel like we're thinking about it the same way, which feels good for me. Anyway.
Man, and I read XK. I have this particular comic for some talks. It's very well chosen. Sorry for interruption.
Okay, thanks. Yeah. So this is gonna be the focus of my talk. So I think data and evals are a big problem, but I really believe that there is an inductive bias mismatch in single cell sequencing, and that cells as a sentence is just not a useful analogy. And I think partially part of the reason why the field has went astray. I mean, that's like a very opinionated thing to say, but has like maybe underperformed is that people have been really captured by this idea because it's simple and easy to model, but I think doesn't really reflect the data. And I'm going to spend a few slides to really hammer home my point of view on this because I think if in 15 minutes I can get people to stop doing this, that would be like really impactful. So let's see. So just high level, thinking about what single cell sequencing data is. Essentially, there's a latent biological space. We all want to learn about that latent biological space because that's where we can discover drugs and understand cells and really understand how to treat patients. And we sample from that latent biological space. We sample single cells through single cell RNA sequencing data. So the data that we observe is essentially the probability of some data X given Z, which is like where we sampled in that latent space. The thing to keep in mind is that data generation process is noisy. So it's contributing dropouts, technical variation. We call that essentially nuisance variation. It's your inability to observe the latent space because you have to observe it through some noisy medium. So, and then just another preliminary to think about, just to define what I mean by cells as a sentence paradigm. So the cells of the sentence paradigm goes something like this. Um, Genes or like cells are sentences, and they're formed by a series of gene tokens. And you essentially want to do mass language modeling on either the gene tokens, depending if you're doing like a ranked gene ordering, or the expression values, depending on what you're masking. You pass that either pair of vectors or single vectors, depending on your setup, through a transformer encoder or encoder decoder. And then you train that model through token level reconstruction using an objective like mean squared error, for example. And then you take your CLS embedding, just like as you would with perhaps language or um, vision transformers, and use that for your downstream tasks. So this is the cells as a sentence paradigm. And I think the key piece here is that you're doing reconstruction on the input in the input space. So comparing text, vision, and single cell RNA sequencing data, let's like try to examine how similar single cell data is to text. So in text, tokens are a really good proxy for your semantic space. And so what I mean by that is if you have a sentence, the words in that sentence induce a distribution over that last token that has relatively low variance. So San Francisco is located in blank. You can have pretty low entropy over what are the tokens that come next. You might have America or California or Northern California, but you relatively know from the context what might come next. In vision, this could be less so the case, where there's actually perhaps a distribution over tokens that's a lot smoother over what might fit in a particular space. So here we have like a sort of a concocted example where you have a picture of a cheetah and like, okay, which of the spots go here? And in single cell RNA sequencing, I would argue that it's much more like this vision example than it is like text. So if you have a sequence of genes, the next gene in that set is really stochastic. There's a high variance distribution over which gene might come next. And indeed, the transcriptional count, like the number of transcriptomes that you observe for a particular cell, is also highly stochastic. There's perhaps a Poisson distribution over that count that's relatively smooth. And in fact, you also have dropout, right? So you have this kind of like enrichment of zeros. So you have, in general, a high entropy distribution over the next token. Another thing to consider about text versus cells is that in text, there's relatively limited input redundancy. So text is information dense in that if you want to predict the next token, you really need to have good specificity about which tokens to attend to predict the next one. So in this example here, right, you need to attend to girl to know that the next token is she, right? It provides the gender. So you have this really nice information sort of causality in some sense, and it's like one token to the next. In masked autoencoding, in vision, this is really not the case. In fact, there's a lot of spatial redundancy. So if you have masking, for example, of this image here, where you have like some sky, you know that most of the patches are very correlated with each other, right? Like probably this patch here, there's probably just more sky because that's how images work. That's how like the world is. And in single cell RNA sequencing, because genes are not organized as sequences, right? They're organized hierarchically in networks where there's co-regulation. Single cell RNA sequencing is really highly redundant. So this is just like a particular cell here where we have a correlation matrix. And you can see there's these large blocks where there are genes that are expressed together. And because of that, if you're focused on reconstruction specifically, models can learn trivial shortcuts to get large, sort of like signal directionally correct. And then combine that with the noise where you have like these errors, which maybe aren't particularly informative, you end up in kind of a problem. So what do I think is the right solution? So and Fabian touched on this as well. I think if you think about two types of models that you might take, or two types of pre-training tasks you might take, you can have a reconstruction task or joint embedding, and you examine those, and this is figures and thoughts as well, drawn from Jan LeCun paper from 2025, I think October, where they actually derive in closed form reconstruction versus joint embedding and derive some rules. It's actually a quite nice paper to read, where they just derive some rules to be able to understand whether reconstruction or joint embedding is going to work better for your particular problem. And what they find is that reconstruction works well in high signal settings where prediction of the input space works and is the signal is high variance relative to the noise. Whereas joint embedding is preferred in settings where nuisance variation is prevalent, and predictions in latent space can avoid reconstructing the noise. So this is used extensively in vision, and I believe should probably be extensively used in single cell as well. And one thing, just like a plug for the field here, I think alignment between noise and augmentation significantly improves learning. We've seen this in vision. This is a really important sort of area of research, and I think it's relatively underexplored in single cell, how to do augmentation of single cells in a way to align it with the noise to be able to do this kind of learning. So there is work, published single cell work that uses joint embedding architectures. I even have the same figure here that Fabian had in his talk, where they're applying these kind of joint embedding architectures for single cell. I think they're more appropriate because of batch effects, dropout, and like donor, which is sometimes noise, sometimes not. We can talk about that later. And there are some example approaches. So SC contrast, SC concepts, and concerto are ones that I found. These are all, I think, contrastive learning examples where you actually have to have, in a batch, you have to have positive pairs where these are things that you want to push together, and negative pairs where you want to push those apart. There are some challenges with these kind of contrastive approaches, although I think they're a step in the right direction. They don't tend to scale very well. You need really large batch sizes for these things to work. And in vision, they've actually moved to kind of these self-distillation approaches that seem to scale a bit better. And that leads me to the work that Johnny and I put together and put out as a preprint end of last year, where here we're taking a model that we call DynoCell, where we're applying Dyno, which is a self-distillation framework that's used very often in vision for single cells. And, I'm not going to go through the details of Dyno here. Suffice to say, we use all the same tricks that they use in vision, like stop gradient, centering, and sharpening. But one of the things that we do in single cell in order to adapt this kind of approach, which I think is really the crux of why this thing seems to work, is that we have two different innovations I think that we've made. The first, and I think the most important one, is this idea of having multiple views of the same cell and having this kind of conditional view. So in perturbation prediction, you want to align a control cell plus a perturbation to some perturbed cell. That's what we call the conditional view. The model learns to align the embeddings of these two different views of the same kind of task. And then we also have this observed view where we're performing covariate matched perturbation alignment, where we have two cells that receive the same perturbation from the same covariate groups, and we align those. And that is a way of using the data to produce the augmentations that are really of interest to us, right? So you have batch effects, you have donor variation. Maybe these are things you want the model to be robust to. You can train the model to align the embeddings in latent space of two cells that receive the same perturbations in the same covariate group. We also implemented a few augmentations. These are really simple stuff. So we applied Gaussian noise, and we have some gene masking. You can think about doing gene masking in different ways. I think you could mask genes as like modules, or you could do larger masking, smaller masking. These are experiments that we've done in the paper. And then just some results. So we apply DynaCell for perturbation prediction. We show that the model outperforms baselines in these kind of single perturbation prediction. That's true for low effect perturbations as well as high effect perturbations. But I think most importantly, we show that the model is able to capture non-additive effects of perturbations from data that is only essentially single combination. So it's never seen this combination before, meaning that the model is able to learn these kind of epistatic interactions. And that's where I think you could see these deep learning methods really outperform the additive or context mean baselines, which is, ultimately these epistatic interactions, these are the things that actually matter, I think, because ultimately these might be like where you could find good drug targets. So here we could show that for FOXP3 and JAK3, which are in the same pathway, so they epistatically interact. We have a high correlation for the DynaCell predictions to the actual observed changes induced by the perturbed cell. So each point here is a gene. And then you can see the context mean doesn't do very well. The additive model doesn't do well at all. And you can see why the additive model doesn't do well, because if you look at the singles, you see that these don't additively combine to create the combination. Okay, so I think I tried to stick to 15 minutes. We have 15 minutes, I think, for discussion. But just to summarize, in conclusion, there's really three different areas that I think, as a field, we need to improve in order to have these single cell foundation models really have the impact that we want. First is data. We need like denser data in local areas that matter. Second is benchmarks. We need really supported and agreed upon benchmarks that are sort of like CASP style benchmarks rather than benchmarks that maybe go a little too fast or aren't sort of supported by the rest of the field. And then I think in modeling, I really believe the field should focus more on joint embedding, where we're doing latent embedding rather than prediction in the input space to avoid these kind of like nuisance variations. So yeah, that's my talk. And yeah, happy to go to discussion.
Thank you, Amaro. Yes, you're right. We have roughly 15 minutes. That's made this punchy. I try to summarize the questions into some like three main questions. I hope we can go through all. So the first is around data. And all of you have shown that your models are getting better if you can input more data and you have more parameters. But there is obviously a limit to that, also in compute, but also how much data we have. So one question I would pose first to Fabian and to the others, what do you think? Is it about more data from a certain type? Should we have more like spatial transcriptomics or single RNA-seq, or is it more diverse data? And the second part of the question is, are we close to an AlphaFold moment where you don't need more data, you need better algorithms, or we are far from that and it's still about the data and not the model?
I can super briefly, but I think then Amaro take it up, in particular Ron with these like huge data sets. But I agree with what Amaro, I think, sort of very nice to say with the tip. We discussed very early on a human cell atlas, when do we stop? And I think we can stop with just getting sort of generic PBMCs. There's maybe enough for that, right? But if you add suddenly genetics, whole bunch of new questions, and then you might need that. I'm super excited about perturbation. There was also a question in the chat. I think with assays such as parse and there's now also related ones that don't just perturb-seq, but can really sort of batch things and add drugs and so on at scale from 10x and others. There's now the way how we can really get many samples. So basically sort of samples in perturbation space and not just cell space. And many of the metrics actually just more or less pseudobulk averages anyway. So it doesn't really help all that much to add just more cells on the same perturbation, but maybe more combinations. And quite recently, there was a cell system I can put in the chat where we sort of worked a little bit with. He comes up with techniques where he can like take each cell as a mini batch for random combination, not just as with CRISPR, but in this case for transcription factor overexpression. And suddenly, boom, Amaro, right? You sample sort of regions in this space where you won't even get there. Because then this stuff is so noisy, so the noise gets another problem. But I think we need to have more of the piece, essentially.
Mm-hmm. Thanks, Ron.
Yeah, our view is, more, we need more of everything still, but in particular, I think more diversity of data. So right now we have, let's say, we have roughly 1,500 lung cancers. We don't think we need more lung cancers. We think we need more other cancers and more non-cancers. And what we see is the models get a lot better as you add a diversity of data. And then I'm very interested. So we think spatial is really important because because biology is spatial and there's like spatial nuance. I'm very interested, and we haven't done this yet, to understand whether we can, whether if you take a model pre-trained on all the spatial data, whether it can infer like some of that spatial context from the single cell data. We'll think about doing something like that in some number of months, but I think that would be really interesting, because then if you can sort of use this spatially pre-trained model to unlock a lot of the single cell data that's out there, then that can be really powerful.
Amaro?
Yeah, so I agree with both comments. I think spatial data is really important. I also think generating a lot of combinatorial CRISPR screens is important for being an engineer. You obviously can't sample the entire combinatorial space, which is why we need these models, I think, for a lot of target discovery. But figuring out how to generate CRISPR data in an area that you care about in combination, I think is important. The one thing I'll sort of add is that I think if you're going to build a company or want to do drug discovery in this space, it's not a dry lab. It's not like only a dry lab problem. You actually have to really have the integrated wet lab in order for this to work. And I think you want to have even a spectrum of experimental assays. You want like really high throughput stuff that's like maybe cells in a flask. You want to have spatial things, so you want like organs in a dish or something like that. You want to be able to bridge all of that and have models that learn from the whole spectrum.
And you think cell lines or some more complex tissue?
I mean, I've only worked on so I've worked a lot on like primary T cells for patients, and found that was, I think, helpful. I think if you look at like, I mean, I don't know, maybe I'm biased because I spent a lot of time at the Broad and the L1000 and all of that stuff. We've done a lot with cell lines. I'm not sure how informative cell lines, or how many more cell lines we need to do. I think if you can work with primary patient samples, it's probably best. Um, but yeah, anyway, so yeah, I think the going all the way into that spatial data and patient data, if you can, I think is like really the most exciting direction, probably. Yeah.
Well, thank you. Let's go to the next question, which is around usefulness and how we can basically translate these scores you have shown, like all of you different scores, correlations, accuracies, etc. And all of you have been talking about the utility and how this can be translated to real life questions. And that's, I think, and then we see that in the field is being a problem, how you can translate these metrics into ROI. How can you then define that, okay, yes, that experiment benefited from my predictions? It's easy to show that you have found something novel, but to show that, yes, out of 100, you can pick the top 10 because my model is that good. How do you tackle this question? And I will start with Ron here. And do you have some hints for the right benchmarks for the community to use?
Yeah, I don't have a satisfying answer for you. And like, the problem here is whenever you're in this regime of like models that are doing new biology and you're not like trying to do supervised tasks, like you're like, what is I don't know what the benchmark is. Like how do I, like, I don't know the biology. So, and this was sort of the
Gene
same
set
problem.
enrichment.
Yeah, totally. I mean, like we had the same problem with recursion, and sometimes we're talking to pharma partners, and they're like, "How do we know this works?" And then they're like, "Well, show me this pathway," and you show them the pathway, and then they say, "Okay, well, how about the next pathway?" And then you show them that way. Like this goes on forever, right? So, I tend to think, okay, like you need to know whether it's working. So like as you start looking at the results, you start to accumulate sort of this intuition, like, okay, if I see things that I expect to see. So for example, our model embeddings capture, there's a group of patients that is like a different histological group. So okay, that's nice. We can at least find that. And then you look and you're like, okay, the patient, now we're finding the PD-1 responder. So we know that that's true. So like I think as you sort of accumulate these examples of the models finding things that they should see, then you can increasingly believe the next thing is hopefully something not both novel and interesting. And then the counterpoint of that is sometimes you do some analysis, and then you look at the data and it just doesn't make any sense.
Mm.
And I think you sort of need to appreciate when you're in that regime of, okay, like I don't think we're quite learning the relevant biology. Obviously we would all love to have very good benchmarks. I don't know what they are yet, but
many of us are looking for that.
Yeah.
Fabian, Amaro?
I would I would say that the way that I think about knowing whether we're going in the right direction, I think the best thing is to get into the gold standard is to get into a lab in the loop cycle quickly, where you actually like use the model to make predictions and then validate those predictions. If you could do this quite fast, you'll be able to sort of navigate your way through these kind of like, is this the right model? Is this the right intuition kind of space really quickly. Agree with Ron though that like that's expensive. It takes like a month. Um, but having really good biologists where you can be like, hey, here are some epistatic interactions. Our model is able to predict these. That's also really useful as well. So I think, domain knowledge and the ability to validate predictions.
Yeah, not super much to add. I think orthogonal data ideally helps. It's you have sort of another modality that didn't use. I mean, Ron, I think you showed H&E maybe sometimes use spatial context, sometimes not. So something orthogonal typically helps you a bit. I kind of like this patient embedding question. I think though one thing one needs to be careful with is right that there's like so many steps until you get your patient embedding. Like, so how do you look for it? Does it sort of drown in the rest? Do you really attend to the right points that you might not see a separation on your desired patients. Your model does something interesting, right? So sort of, if it separates, sure, you're happy, but if it doesn't, maybe not. I mean, maybe that's with any validation. And then the last thing is, and I think this needs to be our gold standard. We really need to do stuff with the models. We need to really build the next experiment, as Amaro said, right? And I think Amaro said it a bit before, that if the assay is cheap enough, right? Maybe it's not just like a single cell assay, it gets like too expensive, maybe too much turnaround. But maybe we just like to find a few FACS features, and have a protein, and then we can really loop it. So we've been trying to do this for multidimensional FACS, and I think it's getting interesting.
And that's something what you can do in vitro, but then how that pairs up with Ron's approach, where there are patient samples and the validation would be clinical. So he can't run a 1 month learning loop. Have you seen signs for like transferring the learning from the in vitro like quick iteration cycle to better predictions in patients?
Yeah, I was just gonna add, it's also very annoying because you're like, well, I have all this human data, and then you want to go run a mouse model, and you're like, well, we don't.
Yeah,
why are we doing this? The mouse model doesn't.
What do you guys think about sorry?
Yeah. I just want to say one thing. This is the fundamental flaw of like all drug discovery, right? It's like how do you go from like things you can tractably experiment on to patients like that? That's like a real challenge, but I think we don't have to invent a new solution. It's just like, just what normal target discovery groups do, where you have like experimental systems that advance in their complexity and expense, and that's how you sort of like get up there. And you still may fail, but
My question is rather if you have seen signs that this can work, because we have seen that in some instances in Turbine, that if we have used in vitro data to predict to in vivo, it was better than without the in vitro. And you can also do that to patients. So if this would work, then we can break this cycle, because you can generate a lot of perturbation data and good models in vitro or lower level organisms, and then translate that into a more complex hierarchy.
I mean, we're seeing this like going from cell lines to organoids, which is sort of one step that's a bit more controlled, right?
Sure, yeah.
I think cell lines, particularly in the homeostatic thing, is like not so much happening. But as soon as we go to organoids and we do something with respect to drugs, suddenly cell type frequencies and things shift that you wouldn't even be able to observe on cell lines, right? So these things, you can really ask more complex questions. You can ask experimental design and so on top of that. So I'm actually quite bullish on this. We have been producing one of the largest sort of perturbed type of experiments in this setting, and also trying to make sense out of it. I think is really fun. Maybe just the second aspect, because before we asked what we could sort of do in terms of validate, I still have the impression in terms of sort of different perturbation types, maybe not yet sort of intersect too much. And it could be that, maybe let's just use a bit more of genetics and like this whole set of perturb-seq things that, quite frankly, people don't really look like, chemo genetic co-embedding could potentially help along these ways. And maybe last thing, Ron, you're looking at images, right? And with sort of simpler readouts, let's say cell painting type of things. And I think you come from recursion, right? So you must know a lot about that. Like there's these co-embedding things in JUMP, for example, one of these big resources, you have genetics plus drugs. So maybe we can sort of learn more about this for validation as well.
Just to add to that, Fabian, I think it's a great idea, this kind of like drug genetic embedding. I think one thing that would be really interesting, somebody should do this, is do some kind of co-embedding where they have the same data set. You give like antibodies or small molecules and genetic perturbations and show that like in that embedding space, you can see that like, okay, this antibody acts on this gene. Yeah, you can see this kind of like genetic embedding aligned with the same functional antibody embedding. Like this kind of thing I think should be very possible and would be really quite interesting to see. It's like kind of an
But is there even, I don't know any screen where people have been using many antibodies, like, like really biologics and as a perturbation. I think that could be kind of cool because then we can do protein space.
Yeah. Ron, you want to generate that for us and put it out online?
So recursion has that data, but it's just not public. And it's really cool. It works. So if you can have an antagonist against the target and you can CRISPR knock out the target and the embeddings like align. So it's pretty.
And JUMP is not enough, right?
What is it? JUMP?
JUMP, this JUMP consortium data, this public one that many pharma did.
Oh yeah, I've not looked at that specifically. So I can't comment on that, but I know like, having seen like a lot of the data behind the scenes, like this works. This theoretic, and a lot of people argue that it shouldn't work. It works. Pharmacology does look like CRISPR knockout if the molecules are selective.
That's reassuring.
Yeah.
Let's switch gears for the final question. And next time we will leave more time for discussion because this is super exciting. And that's about the business model. So there were a couple of questions. And of course, your GSK deal, Ron, is also speaking about itself, how we can translate this innovation into value and ultimately to like working business models. And do you think that that can work only with the data, or it's always or only with the model, or is it always the combination of the data and the model?
Yeah, I could speak to that. So I think it's important. We had a little bit of a LinkedIn debate about this. So it's sort of like it's nuanced. It's like both. Is the combination of data and model. So you need data to train the model. If you have unique data, then you can train a unique model. So I think there's value in that. I think if you've generated a unique data set, then you're in sort of a unique regime. And the problem we all face is, okay, you build these models and you can do a lot of things, and then we don't want to just use them for the one thing that you have the capacity and bandwidth to do as a very small company. And at the end of the day, we're all trying to make better medicines for patients. So it's like, okay, how do we get the most value for these? And so one of the things we were very interested with Kim's group at GSK is like, can we like create some partnership framework where GSK can use the model to support their programs and also with their data as well, because GSK has a lot of unique data that we don't have. And so when you sort of create a partnership like that, then all of a sudden our model working with GSK becomes sort of a unique asset for GSK. It's licensing framework, and then that becomes unique to GSK, but because it's non-exclusive and everyone has different data, then other companies can access our pre-training data and the model with their unique data set, and then you can sort of let the model kind of add a lot of value across the industry. So I think that's we're very interested in those types of approaches, and then obviously we're also trying to do things ourselves. And for us, this kind of question of how to deploy the models, we ultimately felt like we're going to create the most value more quickly if we're using these with clinical samples where they've been trained. And so what we're doing is looking at a lot of clinical programs right now and seeing if we add value there.
Yeah, and that's a good message to everybody who is working on or the data or the models. That the combination makes sense. That there are many companies who can like join forces and make this happen more scale. Fabian, what's your view on this? You are on many SABs, many different business models. You see what is working, what is not working. What's your take?
And I always like talking to colleagues that know this business much more. I have the impression I know the labs and I can very much judge on the papers. I can judge the technical aspects in the startups, but like how they make money with that part, dude, I don't think I can really comment as strongly that I like for one part, for the strongest part, I've been thinking, hey man, platform is the way to go. That's the way to scale. And there's so much excitement. Like virtual cell, we're talking about platforms again. But then the
Sure.
first wave of single cell businesses, I think that sort of aim for the platform model, I think exceptionally all either went bankrupt more or less or sort of pivoted towards a drug development thing, right?
Mhm.
So yeah, I'm maybe more here to learn than really comment.
Amaro, any take on this?
I mean, Ron is inventing a new business model in some sense and being
Right.
successful at it. So I think people should listen to him. But I would say, it's not clear yet, I think, whether licensing single cell models is like or like any model in biology is a business. There are people who are trying and succeeding, I think, right now. Like Chai Discovery or High Discovery is a good example. Noetic is also a very good example now. Um, but I think, ultimately, if you have a model that you feel gives you a differentiation on the ability to discover targets, why not pursue them? Like, that just seems like a way to capture a lot of value yourself. Like, if you have the experimental system and the expertise in a vertically integrated space, um, I would say go build the drugs with that technology. But that's also hard to get funding for now, right? Like, where do you start getting funding for that now? So I don't know. It's not trivial to raise money to do this kind of stuff. It's not. It's certainly not trivial to license models. Um, Maybe Ron can give me some pointers later, but I think yeah, it's those seem like the only two options. Like make drugs or like figure this out. Yeah,
Or combine the two. Yeah, we,
or
we
combine
also
the two.
tried
Yeah.
many of these options, but yeah, we now plan to follow Ron's example with the subscription. Okay, let's close on this. This was a super discussion, very good presentations, a very nice overview. As I mentioned in the beginning, this is our first such meetup, but we plan to continue this. And if you have any like topics you would like to share with us, you would like to hear about, then we will incorporate those. I share my screen just for some QR codes so you can submit feedback or you can continue the discussion on these social media platforms. And yeah, first of all, thank you, Fabian, Ron, Amaro, for the great discussion, preparations, everything. And of course, to the more than 200 attendees on the meetup. It was a nice start. Have a wonderful day, night, wherever you are. Bye,
Thank
everyone.
you for organizing.
Cheers, everyone. Bye.
Bye, bye. Bye.